Hello everyone, today in this video we are going to learn how to analyze next generation sequencing data on Linux command line. The key steps involved in NGS data analysis are the generation of sequence data and FASTQ formatted files, which also include quality assessment of FASTQ data. We see, are the quality scores above an appropriate threshold or not? Genome assembly if required. Sequence alignment to a reference genome, including measurement of read depth and alignment of repetitive DNA. The SAM or BAM format and SAM tools software, which helps in storing and analyzing aligned reads. Variant calling for single nucleotide variants and variant calling for structural variants. We see, are there insertions or deletions, inversions or other complex variants. Then we summarize the variation with the variant call format, VCF format. Then we visualize the next generation sequence data as well as genomic arithmetic with IGV, bed tools and big bed. The final step involves interpreting the biological significance of variants and depositing data in repositories. In this video, we won't be performing all the mentioned steps due to constraint in time and space in my system. We'll be doing the quality control, genome assembly from sequencing data, then read mapping, where we'll map our reads from the evolved line to our ancestral reference genome. In last, we'll use our genome assembly based on the ancestor and call genetic variants in the evolved line. The tools and softwares that we are going to use in Ubuntu operating system are package or tool managing system Conda to install some programs that we'll use in our analysis. It is not installed by default, thus we need to install it first to be able to use it. There are many sources of errors that can influence the quality of our sequencing run. So we'll be using FastQC to deal with the task of investigating the quality and cleaning sequencing data. We'll be using FastP tool to trim adapters and do quality trimming. FastP has a few characteristics which make it a great tool. Most importantly, it is pretty fast, provides good information after the run and can do quality trimming as well. MultiQC is a tool which put FastQC and other tool results of different samples into context. It compiles all FastQC results and FastP stats into one nice web page. Then we'll be creating a genome assembly for our ancestor. We are going to use the quality trimmed forward and backward DNA sequences and use a program called SPADES to build a genome assembly. For the assembly quality assessment step, we'll be using QAST, Quality Assessment Tool, which evaluates genome assemblies by computing various metrics. Then we'll map our reads from the evolved line to our ancestral reference genome. We are going to use the quality trimmed forward and backward DNA sequences of the evolved line and use a program called BWA to map the reads. After mapping, post-processing is done. Here we'll be using SAM tools to fix mates and compress, sort the BAM file into coordinate order and remove duplicates. For seeing the mapping statistics, we'll be using SAM tools and QualiMap, which provides more in-depth analysis of the mappings. For variant calling, we'll be using softwares like Freebase, bed tools, RTG tools and VCF tools. I'd like to share a few research papers for some of the important tools that I'll be using in today's presentation. SAM tools, which have around 33,000 citations, and the paper was published in Bioinformatics Volume 25 in 2009. Then we have BWA tool for mapping the reads. The paper was published in 2009 in Bioinformatics Volume 25. It has around 29,000 citations. SPADES is used for genome assembly. It was published in Journal of Computational Biology, Volume 19 in 2012, and it has around 12,600 citations so far. Next up, we have Quality Assessment Tool QAST, which has around 3,000 citations, and the paper was published in Bioinformatics Volume 29 in 2013. Lastly, I'll be talking about MultiQC, which summarize analysis results from different tools and samples into one report. The paper was published in Bioinformatics Volume 32 in 2016. It has around 1,500 citations. One can take the data from the literature to learn NGS data analysis, or you can retrieve the data directly from the database like NCBI SRA, EMBL SRA or DDBJ SRA or any other database. Once you have the data, we'll download package Tool Managing System Conda to install some programs that we'll be using during our analysis. Go to the Miniconda 3 page and copy the link of its latest version and paste it in the terminal using command wget. This will download latest Conda installer. Bash is the command to run the installer. Different tools are packaged in what Conda calls channels. We need to add some channels to make the bioinformatics and genomics tools available for installation. A channel is where Conda looks for packages basically. Now we create and activate a Conda environment for some tools. Now we can easily install software into the activated environment by using the command conda install package name. There are many sources of errors that can influence the quality of the sequencing run. So in quality control, we aim to deal with the task of investigating the quality and cleaning sequencing data. First, we are going to download the data we'll analyze. Create a directory named analysis using command mkdir. Change into that directory and download the data using command wget and then uncompress the file. Here we are using a paired end sequencing run data from Illumina HiSeq. Thus, we have two files, one for each end of the read. There are a few steps one need to do when getting the raw sequencing data from the sequencing facility: adapter trimming, quality trimming of reads, and quality assessment. Now we'll use FastP tool to trim adapters and do quality trimming. Quality trimming of our sequencing reads will remove bad quality bases from our reads, which is especially important when dealing with the variant identification. We create and activate environment QC and install tools FastP, FastQC and MultiQC. Run FastP using the command shown on the ancestor and then on the evolved samples. Here, detect adapter for PE specifies that we are dealing with the paired end data. Over-representation analysis will analyze the sequence collection for sequences that appear too often. Correction will try to correct bases based on an overlap analysis of read 1 and read 2. Cutright will use quality trimming and scan the read from start to end in a window. If the quality in the window is below what is required, the window plus all the sequence towards the end is discarded and the read is kept if it's still long enough. Threads specify how many concurrent threads the process can use. HTML and JSON specify the location of some stat files. Small I and capital I specifies the two input read files. Similarly goes with the small o and capital O, which specifies the two desired output read files. Quality assessment of sequencing reads is performed by FastQC. We'll create a new directory trimmed FastQC to save the result files. Then MultiQC is used to generate a common report HTML web page having a combined result of all the FastQC files. Here you can see two different MultiQC reports. One is of the raw reads and the other is after trimming. We see that after quality trimming, the quality of the reads have improved. In the raw sequence report, we see that two samples are showing warning for overrepresented sequence. But after trimming, this has been resolved. We observe that per tile sequence quality has also improved. Similarly, by comparing both the reports, we can see what all impact trimming had on the sequence. Now we want to create a genome assembly for our ancestor. We are going to use the quality trimmed forward and backward DNA sequences and use a program called Spades to build a genome assembly. In a recent evaluation of assembly software, Spades was found to be a good choice for fungal genomes. Firstly, we'll change to our analysis root folder and we'll create an output directory for the assemblies. Then we'll submit the paired end data. After this, we'll have to evaluate genome assemblies. We use Quast for that purpose. Install it with the command conda install Quast. Then run Quast with both assembly scaffolds.fasta files to compare the results as shown here. This is the final report of the genome assembly. Here we see the comparative graphical and tabular output for Spades assembly of trim data and the Spades assembly of the original data. And this is the assembled genome and assembly graph with scaffolds. Now we want to map the sequencing reads to the ancestral reference genome. We are going to use the quality trimmed forward and backward DNA sequences of the evolved line and use a program called BWA to map the reads. Again, we'll create an environment mapping and activate it. We'll install tools like BWA QualiMap. BWA is a short read aligner that can take a reference genome and map single or paired end sequence data to it. It requires an indexing step in which one supplies the reference genome and BWA will create an index that in the subsequent steps will be used for aligning the reads to the reference genome. While this step can take some time, the good thing is the index can be reused over and over. Follow this command structure for the mapping. We'll create a reference index for mapping and then run bwa mem. Now that we have created our index, it is time to map the trimmed sequencing reads of our 2 evolved line to the reference genome. We observe there are a lot of header lines. Then for each read, the map to the reference, there is one line. It basically defines the read and the position within the reference genome, where the read mapped and the quality of the mapping. Because aligners can sometimes leave unusual SAM flag information on SAM records, it is helpful when working with many tools to first clean up read pairing information and flags with SAM tools. We are going to produce also compressed BAM output for efficient storing of and access to the mapped reads. Here M adds mate score tags. These are used by MarkDoup to select the best reads to keep. OBAM specifies that we want compressed BAM output from fixmate. Once we have BAM file, we can also delete the original SAM file as it requires too much space and we can always recreate it from the BAM file. This can be done by using rm command. We are going to use SAM tools again to sort the BAM file into coordinate order. Convert to BAM file and sort. Once it's successfully finished, delete the fixmate file to save space. Here, small o specifies the name of the output file. Capital O BAM specifies that the output will be BAM format. Now we will remove duplicate reads. The main purpose of removing duplicates is to mitigate the effects of PCR amplification bias introduced during library construction. This step is not always recommended. It depends on the research question. In SNP calling, it is a good idea to remove duplicates, as the statistics used in the tools that call SNPs subsequently expect this. However, for other research questions that use mapping, like RNA-seq, you might not want to remove duplicates. Let's get a mapping overview now. For the sorted BAM file, we can get read depth for at all positions of the reference genome. Example, how many reads are overlapping the genomic position? Let's extract the depth values for contig 20 and then load the data into R to make a coverage plot for contig not 20 and calculate some statistics of our scaffold. To look at the beginning of X, we use head X. To calculate average depth and standard deviation, we use mean and SQRT of the variance. We'll mark the areas that have a coverage below 20 in red and then save the plot. This is the coverage plot for the contig with highlighted in red regions with a coverage below 20 reads. All these in red shows the poor coverage. For a more in-depth analysis of the mappings, one can use QualiMap. QualiMap examines sequencing alignment data in SAM or BAM files according to the features of the mapped reads and provides an overall view of the data that helps to detect biases in the sequencing or mapping of the data and eases decision making for further analysis. Once finished, we'll open the result page. This will create a report in the mapping folder. This is the detailed statistical report of our read mapping. We can select read pair that have been mapped in a correct manner. Now we'll get the unmapped portion of the reads from the BAM file and extract the FASTQ sequence of the unmapped reads for read 1 and read 2. Lastly, we'll use our genome assembly based on the ancestor and call genetic variants in the evolved line. After installing the necessary tools, we first need to make an index of our reference genome, as this is required by the SNP caller. Given a scaffold or contig file in FASTA format, example scaffolds.fasta, which is located in the directory assembly1, use SAM tools for the same. Furthermore, we need to pre-process our mapping files a bit further and create a BAM index file .bai for the BAM file we want to work with. Let's also create a new directory for the variants. We can call variants with a tool called freebase. Given a reference genome scaffold file in FASTA format, example scaffolds.fasta, and the index in .fai format, we can call variants with freebase and pipe the results into a new file. P1 specifies the ploidy level. We know E. coli are haploid. To understand the output files, let's look at the VCF file and the variants. Now we can use it to do some statistics and filter our variant calls. First, to prepare out VCF file for querying, we need to index it with tab X. We can get some quick stats with RTG VCF stats. We can also run VCF tools to extract more detailed statistics about our variant calls. Now we take the stats and make some plots. We'll do some simple filtration procedures here for both the evolved lines. We can filter out low quality reads. Here we only include variants that have quality greater than 30. Then we'll get the quick stats for the filtered variants. Freebase adds some extra information to the VCF files it creates. This allows for some more detailed filtering. That is it for today's video. Thank you for watching.